everyone, welcome back to our Masterclass Series. I'm Sejal Tapawala, the Marketing Manager at Active Concepts. In today's episode, we're going to discuss the Angiogenesis Tube Formation Assay. And to help us better understand what angiogenesis is and this assay, we have Maureen Drumright, Director of Research and Development here at Active Concepts. Before we dive in, Maureen, we would love to know a little bit about your background and your experience in the industry.
Thanks, Angel. I'm excited to speak with you about the new efficacy test. I hold a Bachelor of Science degree in biology with a pre-med concentration from Queen's University of Charlotte. In 2013, I opted to pivot from medicine to pursue a career in research, and that's when I started at Active Concepts as a research and development scientist. The bulk of my cosmetic industry knowledge started then, when I also became active in the Carolina chapter of the SCC and was elected chapter secretary, chair-elect, and chair in subsequent years. I'm very proud of the work at Active Concepts that I've done and its associated sister companies, from being awarded Best Speaker for a paper presented at a scientific symposium to authoring multiple patents on behalf of our company. In 2015, I was promoted to Research and Development Manager, annually traveled to facilities in Taiwan and Italy to audit and support our international teams, and as of 2020, I've served in my current role as the Director of Research and Development. My focus at AC is supervising all R&D lab initiatives, ranging from regulatory compliance, new product development, efficacy, safety testing, and technical customer responses. Our department goals include producing new and creative products that are supported by robust testing. Before running this novel assay, our clinical validation team researched the topic thoroughly and performed many pilot experiments to screen our materials and validate feasibility. The purpose of this assay is to test products for wound healing capabilities, specifically regarding human dermal microvascular cells, which I'll refer to as DMVECs. We want these ingredients to augment angiogenesis in order to restore skin to its pre-damaged state, thereby accelerating wound healing.
Thank you for sharing. We're excited to have your expertise as we dive into the topic of angiogenesis. To start, can you tell us what is angiogenesis?
Sure. Angiogenesis is the process that forms new capillaries out of existing blood vessels in your body.
That makes sense, as the word angio means blood vessels and genesis means creation. So capillaries are tiny blood vessels, right? What exactly do they do?
The blood vascular network is an important component of the dermis. Capillaries deliver blood, nutrients, and oxygen to your organs and tissues. If some tissues aren't receiving enough oxygen, cells in the affected area send out chemical signals that cause angiogenesis to begin.
That sounds like an SOS call for help. What happens next?
In response, the wound healing process starts with the release of growth factors like epidermal growth factor, fibroblast growth factor, and transforming growth factor from different sources that stimulate epithelial migration and proliferation. These growth factors work in a sequence that starts with vessel destabilization, followed by endothelial cell migration and proliferation to form new blood vessels, and finally, vessel maturation to complete the angiogenic process.
And what is the importance of this angiogenic process when it comes to our skin?
Restoring normal blood flow supports cellular viability as it enables the adequate delivery of oxygen, facilitates nutrient exchange, and increases the removal of metabolic waste, all of which are critical for wound healing. Angiogenesis helps your body heal from wounds and perform other vital functions. This process begins before you're born and it continues throughout your life.
And are there different types of angiogenesis?
Yes, there is sprouting angiogenesis and splitting angiogenesis. Sprouting angiogenesis is when sprouts or new growths of endothelial cells emerge from your blood vessel. They grow toward a chemical stimulus such as vascular endothelial growth factor. These sprouts turn into blood vessels that can reach areas of your tissue that have no other blood supply. Splitting angiogenesis is when a capillary forms without sprouting. Rather, one blood vessel splits into two to form the new capillary network. So instead of spreading outwards, endothelial cells move into the blood vessel's opening and form pillars. These pillars lead to the growth of new capillary branches.
I see. So there are two different mechanisms of how the angiogenesis process can occur. This all sounds like a tightly regulated process.
It is. Strict control is necessary to make sure that new vasculature is only formed when and where it is needed, and organisms have several off and on switches to facilitate this. If these signals controlling angiogenesis are unbalanced, this can result in the abnormal formation of blood vessels, which can play a role in the pathogenesis of many diseases. On the other hand, impaired angiogenesis can lead to delayed wound healing.
So it's important to maintain this balance between the pro-angiogenic and anti-angiogenic signals.
Yes, this is known as the angiogenic switch. This steady equilibrium is maintained through the activity of cellular signaling pathways, particularly through the activation of growth factor receptors.
So in regards to the Angiogenesis Tube Formation Assay, what does this assay tell us?
This assay evaluates the effect of a particular ingredient or formula to develop tube-like capillary structures in vitro. A more complex dermal microvasculature reestablishes blood flow and enhances wound healing.
And so how exactly is this testing performed?
Human dermal microvascular cells, which we call DMVECs for short, are seeded in a tissue culture flask and grown to 80% to 90% confluency in complete media. Microplate wells are then coated with a reagent, Coltrex reduced growth factor basement membrane extract, and allowed to solidify for 30 minutes at 37 degrees Celsius. Human dermal microvasculature cells are seeded on top of the solidified Coltrex phosphate buffered saline, also termed PBS, and this serves as the negative control. Various concentrations of the test material are diluted in complete media. After incubating at 37 degrees Celsius for 18 hours, 3 images are taken per well under a light microscope with a 4x objective. Total tube length is assessed for each well image using ImageJ Angiogenesis Analyzer software, allowing us to quantify the overall length of the capillary structure.
What type of results can we see when using this method of testing?
The negative control, PBS, typically will reduce total tube length, whereas the test material, if able to augment angiogenesis, may increase total tube length when compared to the untreated DMVECs.
So in response to injury, dermal microvascular endothelial cells reestablish a functional capillary network through angiogenesis to ensure sufficient oxygen delivery, nutrient exchange, and metabolic waste removal. This test method will help us indicate if a test material will increase angiogenesis, which may help to accelerate the wound healing process.
You've got it!
Impairment of wound healing is also linked with the aging process of skin. Instead of the skin healing rapidly, it can progress to a chronic state that will increase the susceptibility of having wound infection and scarring. This angiogenesis process is important to how our skin will look and heal as we age. Thank you, Maureen, for your expertise on this topic, and thank you everyone for joining this masterclass on the Angiogenesis Tube Formation Assay. I'm Sejal Tapawala, Marketing Manager at Active Concepts, and with us today we had Maureen Drumright, Director of Research and Development. Tune in next time as we dive into more in vitro testing. Bye!